My pleasure for today to introduce Dr. Sheng. Dr. Sheng graduated from the University of Science and Technology of China, both with a bachelor and a master, then obtained a PhD in medical physics from the University of Wisconsin-Madison in 2004. I hear myself. You know that the microphone there is off.
It's on
The all. Is the right light. The Friday. Okay. I don't hear myself. I want to check with people remote.
I'll ask.
Okay. So Dr. Gishen was an assistant associate professor at the University of Virginia, and in 2011 he moved to the University of California, Los Angeles, as a full professor and a director of the physics research. 2022 then introduced SEPH, radiation oncology, as the vice chair of medical physics. He has broad research interests in radiotherapy and medical imaging, including treatment planning, optimization, image extraction, processing, national technologies, and radiobiology. He was elected fellow of the AAPM in 2016, American Association of Physicists in Medicine. Is that correct? And his research has been supported by NIH, DOE, and national partners. He has published over 180 peer-reviewed papers and mentored over 20 PhD students. Please join us in welcoming Dr. Gishen.
Thank you. It's truly an honor to be here to speak, and also it's great to be able to deliver the talk in person. I've done too many Zooms. This is a good change. So a year ago, I gave a talk about proton therapy, mostly on treatment planning optimization. Today, I want to focus on a few more aspirational projects that we've been working on related to imaging. So that's why I came up with the title Chasing the Light, because that's exactly what we do for these projects. So in the clinical domain, we work with X-rays quite extensively, both megavoltage X-rays for radiation therapy and the kilovoltage X-rays for imaging. So most commonly with the therapeutic X-rays, when the X-rays interact with the tissues, the questions we ask most often is, what is the energy deposited, right? Then once we figure out the energy deposition, we calculate the radiation dose and we correct or we connect it with some sort of a biological effect. And then we stop there. For kilovoltage X-rays, for imaging purposes, the question is, how many photons we send in and then how many photons we get out, right? What is the attenuation along the tissue? The exit signal is then used for either 2D X-rays or 3D CT reconstruction. So the question I want to ask today is slightly different. We want to look into the secondary particles generated from that process and see what kind of information we can extract. So this is a kind of a digital art. My daughter created kind of to illustrate the idea, which is a pair production tomography. So we know that when megavoltage X-rays interact with tissues, there are three primary interactions. The first one is the photoelectric effects, and then the second one is the compton scatter, and the third one is pair production. So when the megavoltage X-ray energy is higher than 1.022, then there is a probability of generating the pairs. The higher the energy and the higher the probability. So the idea was that can we use that to generate the images? Once we have the pairs produced, and then if we have a detector set up very similar to our PET-CT scanner, and then we can pick up the pair production signal, then we can reconstruct. The first idea came through our mind is then we can reconstruct the location of the interaction. Then we can use that to help us determine the radiation dose delivery. So one question is, when we use microvoltage X-rays to irradiate the patients, we generate a lot of signals, not just the pairs. So when we simulate the interaction with Monte Carlo code, you can see that there is only a small blip at the 0.511. And it's easily overwhelmed by the other signals, mostly from the scattered photon. But when we apply a energy filter, so we look into the specific energy window, plus minus 10% of 0.511 MeV, and then you can see the size of the peak more prominent. And then if we apply a timing window, because these pairs are synchronized, if we apply a timing window of 300 picosecond, which is achievable with today's PET technology, you can see that the background noise is significantly suppressed, leaving a very nearly pristine pair production signal. So the question is, how do we reconstruct the images with the signal? There are a few assumptions, a few different methods to reconstruct the images. We can use a filter back projection, assuming events along the coincidence line, filter back projection with an ultra-fast TOF, time of flight detectors, or reconstruction-free method with triangulation. So I'll go through these different methods. So the first assumption is that we assume that we irradiate the patient or the tissue with a broad beam, so it's a volumetric excitation. And then we assume relatively slow detectors, 300 picosecond detectors, and then we do a filter back projection to acquire the image. And what you can see here on the lower right corner is the reconstructed image, and you can see it's very grainy. This is by the way, with about 2 centigrade of radiation dose to the phantom. Above that, you can see the ground truth image with different inserts, iodine, beryllium, gadolinium, ytterbium, 5% concentration, as well as bone, air, and water. So, however, if we have an ultra-fast detector, with 20 picosecond time resolution, then the signal to noise resolution can be significantly improved because of the suppression of the non-coincidental photons. But 20 picosecond detector is very hard to achieve. We are getting there, but we don't have that yet. An easier way to achieve that is actually with a scanning pencil beam. So on the right side, I'm going to play a movie. This is a multi-leaf collimator. I know it's the multi-leaf collimator. Actually, recently we acquired the high-speed multi-leaf collimator. So the multi-leaf collimator is a device with a large number of tungsten leaves that we can chop a broad beam into small segments. So it's making a letter, the word celestial, which is the name of the company. So with this device, we can move across the field, and we only open a small window at a time. And by doing that, we know exactly where the excitation or the interaction event happens. And then we can triangulate when you have the pair production signal, and then you know the beam, and then the intersection of the two lines is where the event happens. Now, with the scanning pencil beam, we can apply also fairly clean hair production tomography images on the lower right corner with a 300 picosecond detectors. So the first application obviously is to see the dose. Now this is a kind of a holy grail in radiation therapy. For many years up until now, we are delivering the radiation dose according to what we model. We trust our model, but we don't have a direct feedback loop of what exactly is the delivered radiation dose to the patient. With this pair production tomography, we will be able to imaging the dose in real time. And you can see that for this particular patient, the reconstructed image agrees with the model radiation dose very well. But I think that's actually the least interesting application to me. The more interesting application is to be able to quantify the Z, because the pair production is linearly dependent on the atomic number, unlike other cross sections. Photoelectric does not linearly depend on the Z, and the competent does not depend on the Z at all. So when you look at the reconstructed images and the quantified intensity of the different inserts, you can see that the CT reconstruction does not really have a clean linearity. And when you have a mixed material, you won't be able to use the CT to derive the atomic number. However, with the pair production tomography, there is a strong linearity with the Z atomic number. So it's going to be great for material differentiation. And what is more, CT is great at many things, but the CT is very limited in terms of showing the soft tissue contrast. This is where we benefited from MR. MR is much better showing the soft tissue contrast. The reason is that for the CT has a large photoelectric component that makes CT very sensitive to the high Z material, but not sensitive at all to the low Z material. But if you think about a imaging modality like a pair production tomography that's sensitive, that's linear to the Z, and then with the very low Z range, which is the majority of the tissue compositions in human bodies, oxygen, nitrogen, hydrogen, they are all in the single digits Z. And the difference with even one atomic number is a huge difference in the contrast, which is showing up here in this for the breast tissue contrast versus the background. And you can see that the CT contrast is within 1%. That's very low. It's easily buried in the background and noise. But with the pair production tomography, the contrast is 13% versus the 1% contrast of CT. So it shows up very, very clearly. You can see it in the reconstructed images, especially this one. This particular insert. I don't know if it shows in the projection, but you cannot see it in the CT. There's one more thing. So what we know that for when we use X-rays to acquire tomography, we need to meet data sufficient condition. So now I'm going to tell you about data sufficient condition, but in the second half of the presentation, I'm going to completely throw that away. Just bear in mind. But just let's stay in the classical CT reconstruction domain. When you acquire 3D images, you need to rotate the X-ray source and the detector around the subject to acquire a minimum of 180 degree plus half fan beam angle to meet the data sufficient condition. However, for the pair production tomography, you don't need that. A single pencil beam is going to give you the 3D information without artifacts along the beam path. So you can see that just a single narrow beam is going to be able to attract the material information. with the same information as you scan the entire phantom. So what does it do to us? Essentially, with this method, we can probe the patient, we can perform the virtual tissue coring or virtual biopsy without irradiating non-involving tissue volume. So of course, there are still challenges to really materialize this technology. The first thing is that we set up the experiment in our therapy room with the megavoltage X-rays shooting down, and then a pair of detectors on the two sides of the couch. But the signal collection efficiency with this setup is super low. With a PET, at least you have a ring, and now the modern PET scanners have a longer and a longer scanning volume to improve the efficiency. So right now we are combating with this very low collecting efficiency. So we can go, each time we can only get a few of these pairs. It's not enough to do a lot. And the second challenge is these single photon detectors are super sensitive to the environment. And unfortunately, in radiation therapy room, whenever we turn on the X-ray beam, we produce a ton of leakage dose directly from the linear accelerator head to the detectors. So we need to figure out a way to better shield those detectors to prevent the pile-up effect. Otherwise, you get saturated. So it's ongoing in terms of the experiment. So this is one of the reasons I want to take this project to here, because I saw that maybe there's a collaboration opportunity. So I want to just give a summary for the first half of the project. And also I want to pause in case people have questions here. NV X-ray for therapy produces pair production photons that can be used for atomic number and dose imaging. The location of interaction can be determined by ultra-fast detectors or relatively slow detectors and the pencil beam. Pair production tomography can be used to differentiate the low-Z materials and perform targeted imaging without exposing a large patient volume. So I want to pause here to see if there are any questions. Okay.
Since you're asking for questions, so you envision for point 3 that you're actually using this new approach for diagnostic purposes, not just for therapy.
Yes.
So even for diagnostic, like mammography, could envision using a 6 MeV photon beam to play contrast, even for diagnostic purposes.
Yes. So this actually was the question when we sent the paper to Nature to review, and the reviewer said, yeah, we like the idea, but I don't think it's practical because I don't think that we have so many MV beams available. I told the reviewers, yes, we do. We have a lot of those MV beams available in pretty much any hospitals in radiation oncology. People just don't know.
Yeah, but of course they're being used for treatments.
They're being used
for diagnostics.
Yes, but they can be used for diagnostics if we can develop the imaging instrument around it.
Because again, you really, sorry, I don't want to take up all your time, because right now for the imaging, you need really pencil beam. Like in therapy, you actually have more broad beam. So it's more extended for therapy.
Even for therapy, we chop down the broad beam into small narrow pencil beams. So we can carve the dose distribution.
Right, but typically it's larger than the pencil beam.
It's typically actually just pencil beam.
And you scan, then for each projection you scan?
So for each field, we want to deliver a intensity pattern, right? So if you look at just like a mountain, then the question is, how do we deliver a intensity pattern like a topography, with different heights? Essentially, we first simplify the shape of this mountain, and then we chop it down like a layer cake. And then we group them up into the groups with the same intensities. And then we try to convert that intensity patterns into so-called apertures. And then we use the multi-leaf collimator to form these apertures to deliver the dose. But quite often we have to chop the broad beam to be very small in order to carve very complex intensity of fluence maps. That's why we are building faster and faster multi-leaf collimators to perform that task. The newest multi-leaf collimator can, these individual leaves can travel at 5 centimeters per second.
Just to clarify, for point one, does that mean that we can get diagnostics at the same time of the therapy, or is it?
You can do both. You can extract both therapeutic and diagnostic information during a treatment session, or you can just have a dedicated diagnostic session. For example, for virtual tissue biopsy or coring.
So speaking of the biopsy, are there diseases or pathologies that change the type of disease so that you can, how does that diagnostic work?
So for the diagnostic, for example, one of the MR techniques is called MR spectroscopy. People perform MR spectroscopy to determine the different tissue compositions, which differentiates tumor tissues from normal tissues. So I can give you another example. For example, prostate. Prostate cancer itself does not have a soft tissue contrast compared with normal prostate cells. However, there are a few things that differentiates prostate cancers from normal prostate cells. For example, the choline concentration in the prostate cancer cells is higher than the background prostate normal cells. That's one thing. Yeah, choline. And more interestingly, prostate cell has a function that can enrich the zinc concentration. And once the prostate cells turn malignant, they lose the function to enrich the zinc. So in normal prostate cells, they have a 10 times higher zinc concentration compared with normal tissue. But once they become malignant, the zinc concentration drop back to the background level. So that's another way to diagnose cancer. So if you can quantify the atomic number, you can do many things.
I'm sorry to spit off, but that really grabbed my attention. So prostate cancer cells, at least of some varieties, have this 10 times absorption. Does that make them more susceptible to these zinc-based radiopharmaceutical?
No, they have a 10 times lower.
10 times lower? Okay,
Yes.
Okay.
So
There's
the
nothing
normal
high.
prostate cell has a very high zinc concentration.
I see.
Yeah.
Okay, and just to go on your question, what did you say, chlorine or choline?
Choline,
Choline,
yes,
not chlorine. Okay,
choline.
thank you.
Okay, so I can move to the second part. So as I mentioned that there is a data sufficient condition when you acquire 3D images, you need to acquire many 2D projections for reconstruction. But this is something that we are trying to break here. So mathematically, when we talk about the cone beam CT, the data sufficient condition was first described by this mathematician. His name is Tui. So Tui's condition says that in order to perform the exact reconstruction of the 3D volume, the voxel of the 3D volume needs to be on the straight line connecting the trajectory of the source. So that's why for a cone beam CT reconstruction, the voxels above and below the center plane cannot be exactly reconstructed. And in order to reconstruct that, you have a helical trajectory. So there are some nuances with the cone beam CT. I'm not going to spend too much into that. But cone beam CT is very commonly used in medical field, particularly in interventional procedures. On the left side, this is interventional fluoroscopic unit for cardiac operations. And on the right side, this is a very mainstream radiation therapy machine that we have a megavoltage X-ray source shooting down to deliver treatment. And then we have an orthogonal KV X-ray setup to image the patient. So when you work with these systems, you constantly have a trade-off between the speed, imaging dose, and the dimensionality. These CRM systems, in order to acquire enough projections to meet the data sufficient condition, you need to rotate it. These are mechanical devices. Not only they are limited by how fast they can rotate mechanically, there is a safety concern. So there is an IEC regulation for these open gantry systems. You cannot rotate it faster than 1 RPM. Because if you collide with the patient. So in that sense, you have a trade-off. Essentially, you can acquire very fast 2D images at a static position, or in order to acquire volume, you have to spend at least a minute, which is not real time. It doesn't reflect, for example, these dynamic processes, the perfusion and the blood flow of the contrast and the respiratory motion on the right side. So there are different spectrum of CT acquisition protocols. On the left side, this is ordinary CT. You rotate the gantry, you acquire dense projections of 360 degree for a fully sampled CT reconstruction. That gives you the highest image quality. And in the middle, this is so-called a sparse view CT. Instead of acquiring these densely sampled images, the sparse view CT images only acquire 10 to 100 projections per rotation. And then on the far right side, we have the so-called ultra fast or ultra sparse CT. That the problem is that what kind of 3D images we can reconstruct with 1 or 2 projections. So there are many methods used mathematically to solve this sparse field CT problems. With the algebraic reconstruction technique, regularized iterative reconstruction of complex sensing, and deep learning methods. But the ultra sparse projection CT with 1 to 2 views is both a sparse view problem and a limited angle view reconstruction problem that have not been very well solved. Unless strong structural priors are available, ultra sparse projection CT problems cannot be solved using traditional methods. So, recently a new avenue just opened for us to rethink about the 3D data problem. So when we think about 3D data, we think about different formats that the 3D data are stored. It can be either stored as a voxels. That's something that we are most familiar with, where you have a 3D matrix, and each voxel is one element in the matrix. And then if you are thinking about volumetric data, they're always large, hundreds of megabytes or gigabytes. And then another way is with mesh, polygon meshes to store the 3D data. That's also very large. Some quite often they are even larger than the voxel data. So in order to generate the information to fill these individual elements, this is one of the reasons that you need so many projections. However, recent research in neural radiance fields showed that the 3D data can be stored much more efficiently. This actually was a research done at UC Berkeley computer science. So we look at these highly realistic, detailed 3D images. The size of these individual images stored as a nerve is only on the order of about 5 to 8 megabytes. So if you store it as a conventional polygon mesh method, it typically takes about 15 to 20 gigabytes to store the 3D scene. So it is much more efficient to represent the 3D things with the neural network widths. So this is how nerve works. Essentially, they start with a continuous 5D high dimensional space with the position and the directions. And each time they from the high dimensional space, they can render into a low dimensional space, because what we see actually our vision, what we see is actually 2D. So each time you can render it. And then once you render it, you can compare with your actual image. And then you can solve this optimization problem, because this is a continuous function, it's differentiable. You can use a gradient descent method to solve it. And then you can minimize the loss. And then once you take 10 or 20 images of the 3D space, then you can pretty much solve this optimization problem with a lot of computational tricks that I don't have time to cover today to reconstruct the fairly realistic, highly detailed 3D images. So of course, this has been very attractive to us in the medical domain, saying that, oh, that's really great, right? If you can use such limited data to reconstruct the 3D things, can you use that to reconstruct the 3D images? So this was quickly picked up by another group, and then came up with this project that's called MapNerve, to reconstructing 3D aware CT projections from a single X-ray projection. However, the published the online paper showed a fairly poor 3D reconstruction quality. So you can see on the left side, this is the depth that was reconstructed using MapNerve for a lung patient. And you don't see much. There's not much of depth differentiation with this method. What the method is good at is, once you give the model a 2D view, the model is good at predicting another 2D view from different angle, but it doesn't actually comprehend the 3D object. So we realized that there are two problems differentiating the natural scene reconstruction and the X-ray reconstruction. The first thing is the photon transportation physics is different. Now, X-rays go through, and when you look at the natural scenes, most of the time you're looking at the light bouncing back. So this is a transmission versus reflection. So the physics model needs to be corrected. The second thing is we need to teach the model, the 3D object, this 3D space instead of a 2D manifold. So we modified the nerve and incorporated these two differences and retrained the model. And then we got much better results for the lung imaging. So for example, on the left side, you can see the reconstructed 3D images with different methods for lung cancer patients. In the middle, this is a MapNerve. You can see that the anatomy is all messed up. But with the tomograph, our method, we are able to reconstruct correct patient anatomy, showing patient-specific features, such as a lung tumor here, and also a pacemaker here. All those things are lost in the MapNerve reconstruction. And with two orthogonal projections, the image quality is further improved. And then this is the difference map versus the ground truth. You can see the residual error is significantly reduced. Essentially telling us that with two orthogonal projections, we can get fairly usable 3D representation of the patient anatomy. And there are some quantitative metrics showing here. Much better similarity index compared with MapNerve, and much higher PSNR with one view and two views. But I think from physics perspective, this is not entirely satisfying. It's a little bit gimmicky, right? So what we want to lead to for today's presentation is we want to rethink X-rays and the CT. Okay, So traditionally, when we think about the X-ray, especially the cone beam imaging problem, we focus very much on the primary photons. Like I mentioned, we count how many photons go in and how many photons get out. And then the difference is attenuation, right? But in the reality, there is a lot of scattered photons, and we don't like those scattered photons for imaging purposes. So the problem with the scattered photons is that the scattered photons also arrive at the detectors. And our current detectors cannot differentiate the scattered or primary photons. And then we take them all and then perform the reconstruction. So as a result, now if we only reconstruct the primary photons, and then we get pretty clean CT images. And then if we reconstruct with both scatter and the primary photons, we get significantly degraded CT images. So there was a study, essentially both the clean CT image and the scatter contaminated CT images were presented in front of the clinicians and having them to identify the structures. And you can see that based on the clean images, the different colors are contoured by different clinicians, and they agree with each other. Once they are presented with the scattered contaminated images, the contours went all over the place. So which means that they don't have the confidence actually to recognize the object when the image quality drops. So there are many ways to mitigate the scatter contamination. People can use mathematical method performing deconvolution, iterative reconstruction with the Monte Carlo forward projection, beam blockers, and 1D anti-scatter grade, 2D anti-scatter grade. None of them are perfect. There are, once you improve some aspect, you have to pay a price in some other aspect. So, back in 2016, I came up with this idea. I discussed with a few colleagues saying that, oh, maybe we can do a time of flight cone beam CT because the scattered photons always takes, for the same detector position, the scattered photons will always take longer to arrive compared with the primary photons. The primary photon goes straight line and the scattered photon take a longer path. Well, but at that time, we realized that the detector is not realistic. The X-ray source is not realistic. We need a very, very compact pulses. So we kind of dropped the idea. But then 2020, another group actually published a paper, time-of-flight computed tomography. They performed the Monte Carlo simulation. And actually showed that with this hypothetical ultra-fast detector and also X-ray source with very narrow pulses, they can reduce the scatter to primary ratio from about 300% to 4%. So get much cleaner cone beam CT images. It's not entirely practical, but it's theoretically interesting. So I think that practically there are better ways to separate the primary and the scatter. So this is another project that we are currently working on. We call it the sparse scatter sampling grid. So we make these templates, and the templates have many cutout holes, all focusing towards the X-ray source. And then we have these little tungsten tubes, and then we can insert the tungsten tubes into these holes. And with a template, we can have different patterns. We can have a different insertion density. So on the right side is the actual device. And then as you can see, that we don't need to densely cover the detector area with the tungsten tubes. The tungsten tubes would reject the scatter because they're all focusing to the X-ray source. With as low as 0.07% of the detector areas covered, we can reconstruct the image, pretty much eliminating the scatter artifacts. So this is a more practical way to reconstruct it or reject the scatter, improve the image quality. But I want to come back to the main this project or this presentation. Let's rethink about the X-ray imaging. The X-ray imaging is, I often make this analogy that we have 1 billion, blindfolded soldier I send into a forest. And then on the other side of the forest, I count how many soldiers coming out without any interaction, right? Then that's the attenuation. But then there are many other soldiers actually bump into the forest. And that's why I have the bloody head. So up to now, the effort has been trying to reject them, okay? But how about, they actually have some interaction with the forest. They actually carry more information about the forest, but it's just we don't have the tool to understand the information they carry. So another thing I want to show here is, okay, so let's say, think about a phantom with a dense object. And with the primary photon only, when the dense object moves along the beamline, the exit signal, there's no difference in the exit signal. The attenuation is going to be the same. So this is why there is no depth information from a single projection, right? However, with what happened? Okay, with the scatter photons, the scatter photons actually are sensitive to the depth of this object. That's reflected in the timing of these scattered photons arrive at the detector. So if we query that information in an optimization problem, in here, we formulate this optimization problem as a function of both the space and the time of the photons arrive at the detector. So there are some mathematical details I'm going to skip. Then we can actually reconstruct physically the 3D information of a phantom without machine learning. So this is a experiment based on Gantt-4 simulation that we have a phantom with 3 cylindrical inserts, dense bone, water, and muscle in a line. And then we create a single projection and collect the detected signal. Here we make the problem a little bit easier. We have 6 detectors in a cube to collect the signal from both the primary and the scatter signals, and then we perform the reconstruction by solving this optimization problem. So as you can see that based on this single projection, we can actually reconstruct the 3 inserts. Although the shape is not quite right, there is some geometrical distortion, but it does give us the 3 distinct objects. This is reconstructed image versus the ground truth image. And if we increase the number of projections from 1 to 6, we can reconstruct a more geometrically correct phantoms for the 3 cylindrical inserts. Although the density is not quite right, so there is still a work in progress. But it shows that even just based on the physics alone, by interrogating the individual scatter photons, we can learn much more about the depth of the phantom besides the XY plane. So I'm going to summarize here, just to kind of wrap this up. There's a lot of things in this second half. 3D combing CT is reconstructed from 2D X-ray projections. The number of projections can be reduced to a few with either machine learning method or with interrogating the scatter photon. CT reconstruction with 1 to 2 views can be achieved with the tomograph, a nerve model with physical X-ray transportation and a 3D supervision. And the depth perception can also is also encoded in the scatter photon. And naturally, we could combine the machine learning method with the 3D or with the scatter photon signal to even improve the reconstruction result. So I'll stop here to take any questions. Maybe I'll just go back to this summary slide.
something else, I can start again. The nerve, again, you have some processing ultimately you're doing in terms of reducing the data. Some filtering, which is, of course, then how to optimize the filter to represent the data, ultimately reduce the data. Like when you say the rendering is kind of in some of this, like a filtering process. How to adjust that, like to ultimately not to lose some information, but not the critical information. So there must be some parameter to optimize kind of the rendering filter, right?
Yes. Yeah, so this is where nerve is extremely ingenious. If you look at how they implement that, they have, because when you render a 3D scene, you want to use as little data as possible, but you want to capture both the large scale features of the 3D scene and then also the granular features, the high resolution features. So they have this multi-resolution sine, the cosine encoding built in. That's one mechanism. Then they also have the multi-scale modeling of the essentially a ray tracing process. So they want to use a very coarse resolution for the part of the thing there's nothing, and then they use a higher resolution for the part of the object that has a lot of features. So those are the two engineering tricks they build into nerve. There are a few others. It's very smart.
But really using the object itself, how well they need to sample. And you're using ultimately changes to really sample that accordingly to adjust again the different scale frequency. No, that's now in the third point. Yeah, it's really interesting that you're looking specifically on, again, not really transmission here, but especially looking at scatter and your projection around it.
Yes.
Like that's really the focus where you really have now. You look at projection where you don't really have the transmitted beam, but really the scatter beam exclusively.
Yes.
And using that information.
Yes.
Which is, it's good, it's great. But I also want to mention that in that regard, using the power of single photon counting. Of course, single photon counting enables a lot of additional features and improvement.
Yes.
Like if you, for example, time of flight, with like hot paper, like of course that's all based on single photon counting. Like otherwise you don't have time of flight.
Right.
So therefore, the single photon counting is necessary for time of flight.
Yes.
But then it's the same 10 picosecond challenge. Like if you want to resolve something in 1 centimeter or millimeter, you need 10 picosecond.
Right.
So that's certainly a couple of problems there too. But again, in terms of the scatter suppression, if you can count photons initially, you can suppress it. But so again, that's in terms of not making use of it, but at least elegantly suppressing your scatter if you can count single photons initially. Like that's one of the advantages.
Yeah, I think.
But again, your point is you want to make use of scatter, like therefore you don't want to suppress it.
Yeah, I think suppressing the scatter is less exciting. I think much more excitingly is how do we significantly improve the image quality of the current city images? I think if we can interrogate the individual photons, then we have 1,000 times more, even more information for us to reconstruct the image, that we can push the resolution from current 1 millimeter, typically medical image resolution, to 100 micron or even higher resolution.
Because you need a side projection, you're not really using single photon, you still have to integrate still, right?
Yeah.
Which is,
We have time for one last question.
maybe in the moment question.
Oh, is there any question remotely?
No.
No question remotely. Yeah. No, let's thank you, Sheng, again.
Thank you.
Well, students are welcome to stay and chat with us.